start this session. This session will be presented by Professor Cesar de la Fuente. He is a Presidential Associate Professor at the University of Pennsylvania, where he leads the Machine Biology Group. He's one of the youngest tenure professors in the history of Penn Medicine. He completed his postdoctoral research at the MIT and earned a PhD from the University of British Columbia. His research goal is to use the power of machines to accelerate discoveries in biology and medicine. Notably, he pioneered the development of the first computer-designed antibiotic with efficacy in animal models. His team was also the first to find therapeutic molecules in extinct organisms, launching the field of molecular de-extinction. Molecular de-extinction has already yielded preclinical antibiotic candidates such as neanderthalin, mammuticin, and elefacin. Professor De La Fuente is an NIH investigator that has received over 80 national and international awards. He's a highly cited researcher in top-tier journals, secured multiple patents, and has given many lectures, including at TEDx. Today, Professor De La Fuente will talk to us about the use of AI for antibiotic discovery. Thank you, Professor De La Fuente. Please, now it's your talk.
Hey, thank you so much. Can you hear me okay? Let me share my screen here.
Yes.
Okay. You can see the full screen.
Yes, now it is.
Okay, perfect. Yeah, so I thank you for the invitation. I thought today I would tell you a little bit about what we've been doing, applying AI methods and computational methods for antibiotic discovery. So throughout my whole career, I've been very much motivated by this huge global health problem of antibiotic resistance. And it's really one of the most underinvested areas in healthcare that kills or affects the most people in the world. And currently it's these bacterial infections, they're associated with about 5 million deaths per year. And by 2050, it's going to be 10 million deaths per year. So it's really a huge global health challenge. And as you can see here, it's estimated to actually surpass every other major cause of death in our society, including even cancer. If we do a quick calculation, those 10 million deaths, they correspond to about a death every 3 seconds. So it's really a huge problem. And so throughout my whole career, I've been trying to come up with out-of-the-box or alternative approaches to be able to counter these increasingly resistant bacterial infections. And so years ago, I proposed with my team a very fundamental question to try to completely change the paradigm for antibiotic discovery, and we proposed to use AI to do this, right? And we wanted to go beyond the conventional paradigm for discovering new antibiotics, which is basically a very physical process. Typically, scientists go around nature and they take water samples and soil samples, and then they try to purify active compounds present in all of that organic matter. But as you can imagine, this is a very painstaking process that relies on trial and error experimentation, and oftentimes does not yield good results, good candidates. And so instead of doing that, we propose to use AI and to take advantage of the decades worth of biological data that we have at our disposal in the form of genomes and proteomes and metagenomes that have been sequenced over the years. And we propose that by developing the right algorithms, we'll be able to mine all of this information. And sort of the analogy is the algorithms operate like a magnifying glass that is capable of seeing beyond what is apparent to be able to find new molecules that are hidden in all of this complex biological code. And really the paradigm shift was our proposal of thinking of biology as information. And if you think about it, biology is just a bunch of code, right? Just like the code that we use to communicate with each other in the form of the alphabet. In biology, you have genomes, proteomes, everything is just a code of nucleotides or code of amino acids. And so we thought that with the right algorithms, we could explore all this biological data and we could identify new molecules that could help us solve many of the problems that we have today. And so this was the paradigm shift. And a lot of what I'll talk about, it's really applying this conceptual framework. The other motivation for using AI was to really try to accelerate the process of antibiotic discovery. It takes a long time. It can take more than the time that it takes to complete a PhD program to come up with potential candidates. Many years, and it's very expensive. So over $2 billion to try to do this. And so really, with computers, we thought that we could help accelerate this whole process, thereby helping reduce the cost and the time associated with it. And out of all the molecules that we could focus on, we focus on peptides, which are small proteins, which we believe they're the ideal training ground for AI, and that's because they have a lot of the prerequisites that are ideally suited for AI problems. One of which is they have an astronomical sequence space, and so we can explore that more effectively with algorithms than in ways that would be impossible for humans to do. Peptides, because they're small proteins, so they're experimentally tractable. We actually have robots in the lab that make these small peptides, and so we can make them in a high throughput manner, and we can scale up, and we can do all of that. So that's also really an amazing property of these compounds. The other thing that makes them really appealing is that they're very tunable molecules. And so by modifying the optimization or the objective function on your algorithm, you can modify what activity you go after. So you can make antimicrobial peptides, but you can also essentially optimize them for other activities like anti-cancer properties or immunomodulatory properties and many, many other things. And so those are some of the characteristics why we think that peptides are the future of therapeutics and why they're really well suited for AI research. And so we started this by actually taking inspiration from image and speech recognition algorithms that we use every single day, all of us, right? Things like Siri and Alexa, things like that. But here, instead of recognizing voices or sounds, we wanted to recognize molecular patterns that were associated with potential antibiotics, right? And so I'll tell you chronologically how we did this. We started by exploring individual proteins to see if we could find hidden within the protein fragments that corresponded with potential antibiotics. So a protein, you can visualize it in 3 dimensions. You can also conceptualize it in 2 dimensions, right? With the amino acid code here on the y-axis. So you have all the code here. So the algorithms, what they do is they run through the code and they identify regions that are predicted to be antibiotics. And these are given by these different colors here. This is all given by this antimicrobial score that we came up with. And of course, this is only a prediction, right? Because it's algorithmically driven. But it's a very powerful prediction because it allows us to explore individual proteins and identify hidden within the protein fragments, for example, this fragment in yellow, that correspond with a potential therapeutic antibiotic. And then using synthetic biology tools, we can extract it from the protein and we can try to develop it into something useful in the laboratory. So we did this for a number of projects where we were exploring individual proteins, but then we realized that we could, with advances in compute power, we could scale up from mining individual proteins to why not try to mine multiple proteins at a time. And that's when we decided to embark on this really monumental journey of trying to mine the human proteome for the first time as a source of antibiotics. The human proteome is all the proteins encoded in our genome. But this was really a massive scaling challenge, because before we were used to just mining one protein at a time, and here we wanted to mine 42,000 proteins. So we thought we had done calculations. We thought the algorithm would take many, many hours to complete this exploration. But to our surprise and remarkably, in just 1 hour, our algorithm was capable of exploring all our genetic information and identify thousands of new antibiotic compounds produced in our body that were previously unrecognized to have antibiotic properties. So this was really amazing, right? That with a very simple algorithm, we could explore all the genetic information that makes who we are, and identify, uncover thousands of new compounds. What we then did is, because in my lab we not only do the computational work and the AI work, but we also do all the experiments. We do all the chemistry, all the microbiology, all the synthetic biology. And so what we did is we synthesized dozens of these thousand out of these thousands of molecules, and we characterized them in the laboratory. We saw that they had antibiotic activity against gram-positive and gram-negative pathogens, so they had this very appealing broad-spectrum activity. And then we tested them in preclinical mouse models of infection to see if they could reduce infections in relevant mouse models. And here we have an example of this is an abscess infection model, where we can see two different experiments. One, the mice infected with Pseudomonas aeruginosa or with Baumani, so two different gram-negative pathogens that are clinically relevant, both of them. And as we can see, treatment with the peptides from our genome and our proteome were able to reduce the infections very significantly. And so this was really amazing to see that we could, again, discover new compounds in our proteome, in our genome, that could then be used as antibiotics. And so this was really the beginning of some of this field. Um, Out of the thousands of molecules that we identified, at least four of them, these four, they're produced physiologically in our body at different levels from low to medium to high, and in different parts of the body, including in the cardiovascular system, the digestive system, and so on. And so it's really, we think that they are playing a role in the immune system, and we think that upon invasion by pathogens, some of these peptides get produced, and they provide a first line of defense, immunological defense against pathogens. So this was just the beginning, but this really sparked a lot of new ideas and hypotheses in my lab. One of which was the fact that we're finding all these compounds here in the human body most likely means that they're not just there. And so we came up with this idea that perhaps we could find similar compounds encoded all throughout evolution and across the tree of life. And that's when we started to we asked the question of, could we find some of these things maybe in our closest ancestors, which are Neanderthals and Denisovans? Could we find similar antibiotic compounds encoded or hidden in their genetic information? And in the process of coming up with these ideas, we essentially invented a new framework for identifying molecules that we call molecular de-extinction, whose ultimate goal is to identify molecules all throughout evolution, to then be able to synthesize them in the lab, to be able to see how mutations or changes that occur throughout time influence their biological function. And we tend to use DNA as the molecule for learning about our past and our present, and but the problem with DNA is that it's not a functional molecule. It doesn't do biological functions, activities in nature. But here we're focusing on proteins and peptides, which are actually the nanomachines that do everything in life. And so we, for the first time, have an opportunity to see how mutations that occur throughout time really influence their biological functions, right? And we can study that. And so this is the framework that we proposed, and we first applied it to ancient humans. And so in order to do that, we developed a machine learning pipeline or algorithm that we call PanCleave, that at a very high level, what it does is it takes all the proteins from Neanderthals and from Denisovans and also from modern humans, which we use as a control. It chews them up, all these proteins chews them up in little fragments, and then we ran two filters, one of which is a machine learning filter, the other one which is a human expert filter, to predict whether those fragments will be antibiotics or not. And I think it's important to emphasize that every important decision that we make in my lab, we take into account the recommendation made by the machine, but also the recommendation made by the human experts in my lab. That really, and I think it's quite telling of really the future of science. I think it's going to be a tight collaboration between machine intelligence and human ingenuity. Um, and so by doing this, we were able to identify the very first therapeutic molecules in extinct organisms. And here I'm showing some examples of some of those predicted therapeutic molecules. I say predicted because this is all machine learning driven. So we then had to validate everything in the laboratory using ground truth experiments. So what we did is we synthesized a lot of these compounds, and we tested them in vitro to see if they could kill bacteria. We saw that some of them could, and then really more importantly, they were able. Some of them were able to reduce infections in preclinical mouse models of infection. So here on the top we have a skin abscess infection model, and on the bottom we have a deep thigh infection model, which is the one that the Food and Drug Administration here in the United States usually wants to see for exciting preclinical candidates. And in the context of these mouse models, the top candidate, the top hit that we found was Neanderthalin. One thing I haven't mentioned is that we had to come up with new names for all these compounds that we're finding because nobody had ever described them before. And so the convention that we're using is we're naming them based on the organism that produces them. So Neanderthalin comes from Neanderthals and so on. And so really, this was the first demonstration of applying the conceptual framework of molecular de-extinction. We were able to identify new therapeutic antibiotics in Neanderthals and in Denisovans, and so really give us the courage to maybe ask maybe a more ambitious question, which was, why not just sample every extinct organism known to science? But once again, when we started thinking about this, this again imposed a scaling challenge, because here we only sampled 3 organisms, right? Neanderthals, Denisovans, and modern humans. And if we were to sample every single extinct organism, that is hundreds of them. So we essentially had to develop a more powerful AI model. And so for several years, painstakingly, my lab, we developed and we trained from scratch Apex, which is a completely new AI model. It's a sequence to function prediction AI model, and what it does is giving a specific, given an amino acid sequence, it's able to directly predict the antibiotic activity of that sequence. It doesn't take into account the structure, which in some ways is a harder problem to actually do it from sequence directly to function, which is what we did here. This is the architecture. It uses recurrent neural nets plus attention mechanisms, and this is essentially it's able to predict activity against many different pathogens. But perhaps more importantly, Apex opened a window into the past, and it enabled us for the first time to sample all of evolution, including the Holocene and the Pleistocene, and sample many different organisms. Here I'm just showing some examples of the ones where we found the top antibiotic molecules, which includes ancient penguins that were extinct in the 50s. It includes ancient zebras, magnolia trees that disappear throughout evolution. It includes woolly mammoths, ancient elephants, and even giant sloths, and many other creatures from the past. And so really, with Apex, we've been able to identify millions of new antibiotic molecules encoded in organisms that people had never really sampled before. And so, just to give you a sense of the magnitude of the exploration, these are all the organisms that we sampled, that Apex sampled. And as you can see here, many of them, we have both the predicted and also the active ones. So we experimentally synthesized and tested them for antibiotic activity. So many of them we actually tested in the lab as well. And finally, the preclinical mouse data in once again 2 different preclinical mouse models. On the top we have the abscess infection model, and on the bottom we have the deep thigh infection model. And just to summarize, I won't go into all this data, but just to summarize, the top candidates came from the woolly mammoth. So that's a compound that we're calling mammutocin. And also many of the top kits came from the ancient sea cow, the giant sloth, and the extinct giant elk. And in this case, some of them, they displayed anti-infective activity that was actually comparable to polymyxin B, which is a last resort antibiotic. So really, some of them were really promising in these in vivo models, and so we're really excited about the potential of taking them to the next stage of development. We're fortunate that this made the cover of Nature Biomedical Engineering, where the editors highlighted this new idea of really trying to find antibiotics in extinct organisms. So you might be wondering why molecular de-extinction. So I'm just going to tell you briefly about what we've learned so far. So first of all, it has enabled us to explore a completely new sequence space that was previously untapped. And so if you actually do a visualization in multidimensional space of every compound that we found by looking at extinct genetic data, and you compare it with every genetic information that we have from today, from the living world today, many of the things that we found, they fall outside of the conventional sequence space. So this is really, I think, very important. So this notion has already expanded how we perceive molecular diversity, how we think about it. And we believe that bringing back molecules from the past, it can help us address problems that we have today. And so if you think about it conceptually, a lot of the pathogens that are killing millions of people today, they've most likely never seen molecules that existed thousands or hundreds of thousands of years ago. And so we're essentially bringing back a whole new arsenal to counter bacteria that are really problematic in today's society, in hospitals, killing many people. Um, but this conceptual framework can also be applied to many other problems, right? We could bring back molecules to try to treat cancer cells or to try to modulate the immune system and so on. And so you can think about it that way as well. So a couple of ramifications from this work that I thought would be important to highlight. One on the bioethical side, when we were initially discovering all these compounds from Neanderthals and ancient organisms, of course, it was exciting scientifically, but then we started worrying. You know, is it okay for us to synthesize these things using chemistry in the lab? Because when we do actually alignment on the computer for many of these sequences, we can't find overlap with anything that is expressed today in biology today, and so we can't really find some of these molecules in living nature today. So we're essentially resurrecting them using chemistry. And so is it okay for us to do that? So we've been consulting with bioethicists to make sure that we innovate always, but that we try to do that in a responsible way. And so that's something that we've really taken very seriously. And then on the patenting side, another interesting point is that when we were initially unveiling all these compounds, I went to the patent office at the university, and I told them that I was aware that natural sequences are not patentable because they belong to humanity. But then I asked them, but what about all these compounds that we're discovering throughout history from the past, right? Can we patent those? And so this is opening up a whole new area of patent law because they don't know, people don't know, and it's quite entertaining to see lawyers fight with each other about whether we should patent or not some of these molecules that we have discovered. So I thought I would highlight a couple of these ramifications from this research. In the meantime, I've written this opinion piece in Nature Biotech on some of these concepts, some of these ideas, and sort of throwing out a couple of thoughts and my opinion on some of these things that I mentioned. In terms of next steps, we plan to mine the world's biological information. We think of biology as an information source that can be mined with the right algorithms, so that's what we're trying to do. And we're mining the whole tree of life as a source of antibiotics. That's just the beginning. We then are going to go beyond antibiotics to other types of therapeutic molecules. But since we had first started with eukaryotes in the form of modern and ancient humans, we next decided to see if we could find antibiotics in another huge branch of the tree of life, bacteria. So I'm going to tell you about two collaborations where we looked at microbes as a source of antibiotic molecules. The first one was this one where with machine learning, we sampled what we call the global microbiome. These are almost 90,000 microbial genomes and over 60,000 metagenomes that come from all around our planet. So you can see here from many different sites, including oceans, soil samples, oral cavity, cats, dogs, corals. Like it's really an amazing representation of the microbial diversity on our planet. And with machine learning, we sampled all of this genetic information. And amazingly, we're able to identify almost 1 million new antibiotic compounds encoded in all of this microbial dark matter, which was really incredible. We have released all of this data publicly to anyone that wants to access it, fully open access, in a website that we call AMP Sphere. And we did that in an attempt to unite the scientific community so that amongst all of us, we can synthesize some of these compounds and we can try to develop them into, the next antibiotic, hopefully, that can help people. We then, out of those almost 1 million, we synthesized 100 of them using chemistry in the lab, and 79% were active in vitro. And then in vivo, in the preclinical mouse model, a couple of them were highly active highly effective at reducing infections. Again, we had to come up with new names for all these molecules that we're identifying for the very first time in science. And just briefly, the other collaboration was with our colleagues at Stanford. And here we wanted to look at human microbiomes as a source of antibiotics. And the premise for this is that we thought that human microbiomes would be a great place for innovating at new chemistries and new types of molecules. And that's because these microbes, they live in communities of millions or billions of them. And so they have to fight each other off to take over a particular space and then be able to grow into microbial towns and then microbial cities and then microbial countries. And in the process of doing that, we hypothesized that they would produce molecules that would help them fend off microorganisms that are in their surrounding areas. And that's what we found. So we sampled almost 2,000 human metagenomes, and we identified individual molecules that are produced by individual microbes that they use to kill off the surrounding microbes. And so this was really, and here are some examples of that. We found hundreds of them. And then we tested them in vivo in two different preclinical mouse models, the same ones that I described before. So the skin abscess infection model and the diphtheria infection model. And I won't go into all the data here, but this was a lot of work, a lot of mouse experiments. But the top candidate was a molecule that we call prevotellin-2 that is produced by the gut microbe, Prevotella copri, that each of us has in our intestines. And so this is another great reminder of the really amazing capabilities of microbes to produce new molecules, right, that could be helpful for humans. In this case, to treat bacterial infections and so on. So we're excited to try to continue to do this work, sampling microbes as a source of new molecules that might be useful. And just very recently, we posted this preprint just a couple of weeks ago, where we mined the world's archaea for new antibiotics. So that was the last branch of the tree of life that remained to be sampled by our team. And we're able to use, again, deep learning to sample all archaeal samples known to science. And we found a new class of antibiotics that we call archaeacins. And many of, I won't go into all the details here, but several of which were effective also preclinically. So this was really cool to see. As you probably know, archaea are really underexplored in general in science, but especially so as a source of molecules. So this was really, I think, innovative in that sense. And a couple of these very recent, fresh papers. The other one is Apex Go. This is a new generative AI model that we've developed to optimize antibiotics, and it uses a transformer deep learning architecture with Bayesian optimization. And so basically what it does is it takes a template antibiotic. In this case, we've used some of the extinct peptides, including from the woolly mammoth. And then in silico completely, it's able to take it and then optimize it and generate a lot of variants by sampling through sequence space that are predicted to be a lot better. So it's able to reduce the MIC. So really making them, optimizing for antibiotic activity. And this is really the first ground truth experimental validation of generative Bayesian optimization in any setting. So this was really exciting. We have a preprint on this in case you want to check it out. But again, this is just the beginning. I think the algorithms like this, we can use them to really optimize any molecule to then for a particular function, in this case, antibiotic activity. And the other recent paper is that we're all familiar with multimodal models like Gemini or DALI that can do multiple things, right? They can take a text and convert it into an image or convert it into video and things like that. But we have never applied multimodality to molecules. And so here we just finished, we just put in a preprint for this. But basically, we've developed this model called Apex Duo. It's a multimodal model. capable of designing molecules that can do two functions. In this case, we decided to go after intracellular infections. So most antibiotics that we have out there today, they just target bacteria, but once bacteria hide inside human cells and they replicate inside, the antibiotics that we have, they cannot actually penetrate into mammalian cells and target these intracellular infections. So we decided to use AI to try to build a multimodal molecule that could do both things. They could penetrate into mammalian cells and then target the bacteria inside the cells. And Apex Duo, I won't go into all the details. We have a preprint, but it was able to do that. It was able to create some molecules that could do both, and we experimentally demonstrated this in a number of experiments, both in vitro and in vivo. So just to sort of summarize, right? With traditional antibiotic discovery methods, you can take many, many years to come up with a candidate, which oftentimes it fails subsequently. Today, in my laboratory, in a few hours on the computer, we can discover hundreds of thousands of candidates, and we can go from discovery to synthesis in about a week. So really, this is for all the hype that AI has. This is already a huge success, I think. And on any given day in my lab, I come in the morning. Like this morning, I had a cup of coffee, and I know that by lunchtime or by dinner time, we're going to have a ton of new molecules to play around with. So it's really like a playground for a scientist like myself, who's been working on antibiotics for many many years, and so it's really an exciting moment. For this type of research. And I think a lot of it has been, of course, enabled by AI. And I haven't been able to talk about all our advances, but here I have just a summary slide in case anybody's interested about all the advances that we've been able to do, the top ones in this field of antibiotics AI, which is really an emerging field. And I've touched upon some of these, but not all of them. And these are some of the preclinical candidates already that AI has discovered in my lab. And this list continues to grow, every month or so. And so the dream that we have is that hopefully some of these make it into the clinic and can help people in the future. I mean, our ultimate goal is to benefit humanity. So hopefully we can see that. But so we've done a lot of work in the antibiotic field, but really this is just the beginning. I think we're in a new era of peptide design. And what we've demonstrated in the context of antibiotics is that by building good data sets in the lab, we can then use them to train AI models like Apex and Apex Duo and Apex Go that can then do really things that were impossible before. And they can design and create new molecules that we can use, like I mentioned so far for antibiotics, but you can use the same framework of generating good data and then using that as a training set for training AI models. You can use it for many, many other things. So in the future, I'm excited about the prospects, and I'm excited about applying AI and peptide design in fields like immunology, and we're already doing that, neuroscience, food science, agriculture, livestock, oncology, and many, many other fields like materials, tissue engineering, diagnostics, and we're collaborating with people to do some of these other applications as well. So it's really an exciting time for peptide design enabled by AI and by data and automation. And with that, I'd like to thank lab members, past and present, our amazing collaborators from all around the world, our funders that allow us to do the work that we do. And here I'll just leave you to links to our website, my email, and social media, in case anybody wants to anybody wants to get in touch. And yeah, so thank you so much for having me. I would love to take any questions that you might have.
Thank you, Professor De La Fuente. Excellent presentation. And now I'd like to open for questions. Well, while the audience thinks, I'll have a few questions myself. Well, this very extensive data set you have, and you developed a system that can search for antibiotics. But would it be possible to consider interactions or for synergism in this system to search for possible interactions from different kinds of antibiotics?
That's a great question. So we're actually working on that. We have a model that can predict synergistic interactions between peptides and between peptides and small molecule antibiotics. So we are working on that at the moment. It's not fully ready yet for prime time, but it's something that we're certainly interested in and working on at the moment.
Great, thanks. Other questions?
Thanks for the wonderful talk. When you discover these new antibiotic peptides, is it your impression that in prehistoric times also the same scaffolds have been used by nature? Because for function, the scaffolds are very important, of course, to have, let's say, an antibiotic effect. So did you discover scaffolds that are extinct nowadays and that you said, these scaffolds were used successfully in ancient times?
Great question. I mean, we have found that when we typically do systematic comparisons with everything that we find compared to everything that has been described in literature from extant biology. And we do see differences both physicochemically and in terms of physicochemical descriptors and scaffolds, and also compositionally. So yes, we have been able to find new families, new classes of peptides that have different. They're actually different from what has been described before, simply by looking at ancient biology. So it's really quite exciting in that sense.
Hi, my name is Célio. Thank you for your fascinating presentation, fascinating work. My question is from someone who does not understand pretty much about microbiology, and neither understand from deep learning, etc. What I'm thinking is that, did you concern about inserting in the algorithm situations related to the physiological environment, like temperature, like pathways that could not be activated by antibiotics? Because thinking on the Death Valley in the drug discovery, one of the main problems for drug approval is side effects. And then with these algorithms and the power of AI, it could be possible introducing these variables while searching for new molecules. Thank you.
That's a great point. We are interested in we are in parallel building different models for things like cytotoxicity and stability and PK/PD profiles and all these things that you need to actually create a drug. The problem is always the lack of data, of experimental data, right? Because we like to create our own experimental data sets. So we are working on all these things in parallel. It's just we don't have enough data for some of them to incorporate them into an algorithm. But the ultimate goal that we have is to really build individual models for each of. So we have Apex, which is an antibiotic predictor model. We have Apex Duo, which can do both cell penetration and then antimicrobial activity. But we are working on similar models for cytotoxicity and all these other things. And the ultimate goal is to then be able to combine them all into a single super algorithm that will be able to do multi-objective optimization, right? To then be able to create molecules that have all of those properties incorporated in them into single molecules.
Question here. La Fuente, congratulations for your presentation. My name is Rui,
Thank you.
São Paulo State University in Brazil. In your opinion, we know that the pharmaceutical companies they like just blockbusters, and the antibiotic is are out of the blockbusters. In your opinion, until what the stage we need to move, we need to develop a new candidate to call the attention, disperse the attention of these pharmaceutical companies in order to develop a new medicine with these amazing candidates?
Great question. It's really difficult, right? Because there's not a lot of investment in antibiotic development, and it's a complex situation. But there's not a lot of incentives, market incentives, to develop antibiotics, just because they're very cheap molecules. So there's not a lot of return on investment for investors. So that's why big pharma has, mostly stepped away from this field, and it's a bit concerning. go, because again, like, the market is difficult, and then it's a bit concerning that it's really upon a couple of academic labs like mine here to try to come up with to try to think about this big problem in an unconventional or out of the box way. So yeah, that's the situation. There's not a lot of money. It's not like in cancer where there's a lot of money, a lot of investment. In antibiotics, there's not a lot going on, and so that's something that I've been thinking about. How can we transition some of these molecules to the next stage? Could it be through a non-profit organization or something like that? So if anybody has ideas or knows somebody that has a lot of money or wants to invest in this, let me know, because I'm in the process of trying to figure that out.
Okay, thank you. Other questions? Other-
Thank you for a very nice talk. But with all this
you.
development of AE, do you think the traditional research will survive, or all of us may observe in this direction? Because it's
Yeah, good question. I
one
think
side is
no,
very
I think
nice, the other side where something said if you don't use AE.
yeah. No, I think there's a place for everyone. I think, like, in my lab, I have a lot of experimentalists that all they do is experiments, right? They don't do any computational work. And I think the best environments for innovation are going to be places where you combine both. And then with automation, and then I think in the future, I think in research labs, I think humans will be left to do the thinking, a lot of the thinking, and designing good experiments and coming up with new ideas. So I think humans are still very much important, I think, in the whole process of science and applying the scientific method. So I don't see, I'm not too worried about a replacement. I think we still have a lot to say.
So have the space for some time at least. Thank you.
At least for some time, I think we're okay. Yeah, we're not going to be taken over by machines.
Okay, I forgot to present me. I am Maria Helena from Santa Casa and the Federal University of Minas Gerais. Thank you.
Very nice to meet you, Maria Elena.
Other questions?
I think, hi Professor, my name is Lucas from University of Brasilia. Really, really interesting data and platforms, everything. I was concerned, same thing about Maria Helena just said to you, but I have curiosity, because I was wondering while you talk, how you came up with this idea? How you you came up with this idea to look 100 years ago, a million years ago? So how came up this idea? Where came from this idea?
Yeah, thank you for your question. So basically, we had found these antibiotics in the human genome. So we just started thinking about this in an evolutionary, from an evolutionary standpoint, and we were wondering whether they were just clustered in the human genome, which is unlikely. And so we came up with this hypothesis that we would be able to find them all throughout evolution. And so since we had looked at the human genome and proteome first, we wanted to see whether our ancient ancestors also encoded for some of these molecules. And then that's when we found that. And then we said, well, let's just do every extinct organism, right? We just became more ambitious as the conceptual thread went along, and we started to uncover more and more information and more insights. But yeah, that was basically it. I think we were willing to think of this problem outside of the box, and then we decided to do the work and the research, and we had the capability to do all the machine learning and all the experiments, and so it really the stars aligned in a way, and we had a lot of fun all throughout the process.
Thank you, Professor De La Fuente. I
Thank
think
you.
we don't have more questions. So thank you again for your excellent presentation and for the discussion of the questions.
Thank you. Happy to, if anybody has additional questions, shoot me an email or through social media. Happy to discuss there. But yeah, thank you again so much for having me. Really a pleasure.